We good? Okay. Hello, my name is Greg Simpson. I'm the founder and CEO of Adult Genetic Editing, and I've been asked to do a brief video explaining why we're here and what we're all about. So here we go. First thing I want to do is take you to a website, the National Center for Biotechnology Information. This is a .gov website, and it has things on it related to DNA. Database has in it a variation viewer. What this is variations in DNA that scientists have discovered and their effects on human beings. And we're looking at, I believe this is chromosome number 2. Yes, chromosome 2. So that'd be the second one. So there's quite a bit of data, but there's a lot more data in DNA than just chromosome 2. If we select this whole thing, see if I can do that. There we go. Something interesting shows up. It's loading the variation data, so it's going through the database and extracting everything that is essentially on chromosome 2. And this column right here, most severe clinical significance. This is interesting. If we come down here and select that, we get only the variations in DNA within chromosome 2 that are pathogenic. Now, what's pathogenic? Pathogenic, causing or capable of causing disease. Okay, so these are things that make people sick, genetic changes. And in chromosome 2, we have 4,408 of them. Yes, 147 pages of sometimes single changes in the DNA. Just a C is in place of a T or something like that. And in other places, a little more complicated change or variance. But what's interesting about this, so there are 4,408 in just chromosome 2. How many are there in the total DNA that scientists have uncovered so far? We have a look at this press announcement from the FDA. We all know who they are. The FDA on April 6, 2017, which is just about a year ago, allows marketing of first direct-to-consumer tests that provide genetic information for certain conditions. And what are they telling us about? 10 pathogenic conditions that cause disease based on changes and variations in DNA. What is the FDA? The FDA is an agency within the U.S. Department of Health and Human Services that protects the public health by assuring the safety, effectiveness, and security of human and veterinary drugs, vaccines, and other biological products for human use and medical devices. The agency is also responsible for the safety and security of our nation's food supply, cosmetics, dairy supplements, products that give off electronic radiation, and for regulating tobacco products. Okay, that's the FDA. Nowhere in this description do I read it also maintains control of the information that you're allowed to know about your own DNA. However, they have chosen to only allow 10 diseases. And back here, we've seen 4,408 only in chromosome 2. Now, if we go to the terms of service for a popular genetic sequencing service, you can see that this thing is quite lengthy. There's a lot of terminology here, most of it legalese. Most of it is very much slanted toward that company and protecting it. Down here in paragraph or group 6, item 6, user representations. This is very interesting to me. You are warranting that you are not an insurance company or an employer attempting to obtain information about an injured person or an employee. Warranting, you promise you're not a bad guy looking in the 23andMe database to gather information behind an employee or an injured person's back. The very fact that this is here shows that someone who is that could get access. And they're depending on promises that, oh, you're not here to do anything that the person wouldn't want you to do with their DNA data. Also very interesting to me is waiver of property rights. Basically, what this means, if your DNA that goes into the 23andMe database gets used and it is the trillion-dollar cure for obesity because you're genetically immune to obesity no matter what you eat, you get nothing. And most people, I don't think, are aware of that. They want to use your information to test biological weapons. You don't have a say because you've opted in to allowing genetic research on your DNA. 23andMe does, to be fair, allow you to opt out. Most people probably don't know the repercussions. I'd like to show you our terms of service. This is all of it. And essentially, I'm not going to read it to you. You can pause the video and read it yourself if you like, or come to the website and have a look. A very huge part of our purpose is to ensure that you are the only one that has access to it. And if you choose to share it with someone else or a researcher or a weapons developer, that's certainly your choice. But we're not going to make that decision for you.
Je veux te voir, je veux t'avoir. New York dans le nightclub, tu t'es rapproché de moi. Je ne vais pas oublier le contact de ta peau, ton sourire et ton rire. C'est comme si on se connaissait déjà quand on s'est mis à danser. Tu m'as donné un baiser, que Cristal avait posé là sur la langue. Fait sonner son top bandit, je crève en voyant sa patrie. Moi dans mon cœur, Baori des pas de Tatiana. Blouson de cuir à la bouche, ce qui fait chaud au nightclub. Elle se sentait si bien qu'elle me...
Okay. So you can see one of the main purposes behind adult genetic editing is your privacy. We take it very seriously, and it's one of the main three issues we're trying to solve. I want to talk a little bit about how we'll go about securing your information so that it's not usable for purposes that you don't intend. How it's going to work is when you submit your sample to us for sequencing, we run it through a machine like this. We flow the DNA through there and electrically detect which molecule is passing through a membrane. And the letters stream out through the USB connection here into the computer, and we end up with your 3 billion letters. 6 billion letters, actually, 3 billion pairs. Now, that's a lot of data. That's over 2 CDs worth of data. So we don't store the entire genome. That would be wasteful of storage space. So what we do instead is, back at the NCBI website, they have a thing called the GRCH38. What it is the most common DNA. Of many people that have been sequenced, this DNA is not averaged, because you can't average a CATG. It might be a F, and there is no F. So what it is the most common. Most people along that strand at that exact molecular location have a C. If you have a T, you vary, and that's a variance. And we would record that variance, its location, and we would store that in a little code that would have the location and the variance. We would also have a unique ID for that change, and basically that unique ID then goes into a file which is encrypted and stored in your DNA wallet. Now, you would have probably on average about 3 million changes. That's about the size of a JPEG. They're about 3 megabytes in size. This might be a little bigger based on overhead and how we're going to store the molecular location, because that can be a fairly large number. So once we get your variants, we will then add into a file all of your variances, encrypt that, and give it to you, and you hold the key to reassembling your genetic information. Now, once we get it stored, you're the only one who can reassemble it. Now, how do we guarantee your privacy? This is the part I like. We're essentially going to take and wait until we have probably 10,000 users. Now, you would have 3 million changes, and then we would have 10,000 users, or maybe, let's say, 5,000. 5,000 people who've had our sequenced, their information sequenced. We would store that offline, off the internet, where it can't be touched, and it would be stored in fragmented, so we can't even put it back together. We don't know what's there. Now, once we have these 3 million changes times, let's say, 10,000 users. Alexa, what's 3 million times 10,000?
3 million multiplied by 10,000 equals 30 billion.
30 billion. Okay, so we have 30 billion pieces of DNA changes that are all jumbled up together and then randomly sent out to the blockchain. If a nefarious user wanted to recreate you, here's what he would have to do. He'd essentially have to reassemble a puzzle. Each individual piece is like a puzzle, and the puzzles don't have any interlocks, so there's no way. They're all square. They all look the same. The only difference is they might have 4 different colors on them. One for C, one for T, one for A and G. So he's got 30 billion squares there that he has to put in the proper order. If he gets one wrong, the puzzle's not done. It doesn't work. It won't be you. Now, what's worse about this is there's no picture on the box. He has no way to know that this person who's doing something bad with your DNA has no way to know he's got the puzzle correct, unless he has all 10,000 people already sequenced, so he has the picture of the box. What's the point in doing the puzzle? All right, that's how we're going to secure your data on the blockchain. Now, what do we do with it? Well, one is we're not going to tell you about 10 differences. We're going to work out a way where the blockchain has code. I'm not saying we're going to write this. I'm saying that it would be very easy for someone to write code that would then take these variances and start to find patterns in them and report back to, hey, I found a pattern. Now, the community we believe that we're forming of DNA owners that are part of this DNA ownership on the blockchain group would make the decisions as to how they wanted to be informed and what they wanted to be informed about. Once the software is running, it would be capable of finding patterns in the DNA and reporting back to end users about those patterns. This will make it rather difficult for anyone to tell us what we can and can't know about our DNA if the blockchain is running the code. So, unless they shut down the internet, theoretically now you could get reports back about your DNA and what's in it and what's not in it. Now, certain genes are going to be problematic for people, and certain genes are going to be, woohoo, I got it. I got the FOXO3 that means I'm likely to live till I'm 90 without any significant aging taking place, mentally or physically. I'll be healthy and I'm going to have a great long life. Now, if you wanted the FOXO3 gene turned on in yourself, you would do something called CRISPR, and you could turn that gene, if you found out if it was off, for example, you could flip it back on. And CRISPR is pretty interesting stuff. This is a MSTN knockout. What it is it turns off the MSTN gene in humans. And it's right now floating in a little vial of bacteria, so that if we feed it some sugar, we can make more of this stuff for very, very inexpensively. And this little CRISPR editing device, it's a molecule inside a protein, and that protein will roll on my DNA when I take this and find that spot where the myostatin gene is and cut it. And that will effectively knock out that gene, so that gene won't be doing what it's supposed to be doing. And what it does, the MSTN gene, if you've ever seen those bulls or those dogs that are buff beyond buff and they have muscles upon muscles, well, they have no myostatin. The myostatin gene in those animals has been knocked out. So, and there are humans with that as well. It's a genetic variant, and when you don't have the MSTN gene turned on, your body doesn't know to stop making lean muscle and start making fat and make weak muscle because you're aging. So the MSTN gene could be taken, the CRISPR edit could be taken to turn that back on so that it would be easier for older folks to create lean muscle. And then if they got too big, they would basically just do another edit, which would then knock back in that MSTN gene. It would turn it on and it would go to work, and then they would age naturally over time. But there's a way to control this, is my point. Now, that's one gene. There are multiple things that we know about. The HNM5 gene is present in a lot of Northern Europeans. And I believe if they're both knocked out, those people are essentially very, very, very resistant, almost immune to AIDS. And what that means is that the N5 gene is the creation of a receptor for the AIDS virus to attach to. And if they don't have that receptor and their bodies don't make it, the AIDS virus can flow right through them and it will never actually attach itself to the body and they will not become infected. We believe that there are many more of diseases that are similar. Malaria is one, and they do know that there are people without receptors to malaria, and that is a genetic base for that. Of course, it's not on the FDA's list, so most folks don't know about it. So we've covered, I think, pretty much what I wanted to talk about, how we're going to store your DNA so that it's safe. Talked about your privacy. That's very critical, more so than you know. And in the future, as things evolve, to be able to edit DNA, and that becomes more common in the world, which it can't not. We humans don't necessarily look at new technologies and not embrace them if we see benefits to ourselves. So I do expect that there will be lots and lots of things out there that are changed through the CRISPR technology now that it is affordable, reversible, safe, and effective. So that's CRISPR, that's storage of the DNA, that's the community. I think the last thing I want to say about the community is that we will treat the community like our boss. We at AGE really feel that we're effectively nothing more than a vendor. And our role as your vendor is to sequence your DNA and store it securely on the blockchain. And at that point, our relationship is over with your DNA. Now it's yours, and the community will make the decisions as to how that gets used or not used, and for what purpose that data is there. I want to thank you for your kind attention and hope you have a wonderful day. Hope to see you again. Thank you. Bye-bye.